Hello again, Jared Pitts here, and today we're going to be reading another article by Dr. Ray Peat. This one is called Leakiness, Aging, and the Big C Word, which I try to avoid saying because on particular platforms, most of the scenarios in which Dr. Peat would use that word, it would be frowned upon, to say the least. Okay, before I get into the bulk of this article, just want to remind you that you can visit my website, MissouriHealthCoach.com, and you can set up a free 15-minute consultation to see how I can support you in your health journey. I also have several links that use my code for a couple of different companies like Health Natura and Life Giving Store for their desiccated thyroid, aspirin, vitamin K, vitamin D, and other things of this nature. Things that Dr. Peat researched and shared with us about. If you use my links, you'll get a certain percentage off, and it will help support the channel. Without further ado, this is how it goes. A thin layer of fibrin lining blood vessels provides a filtering barrier that helps to strengthen the wall and prevent other proteins from leaking out of the vessels. And it participates in repair processes when the blood vessel is broken. Cellular energy metabolism is the basis for maintaining the barrier functions. Energy depletion causes the endothelial cells lining blood vessels to become excessively permeable. When the organism's resistance is low, proteins and fats that normally remain inside the bloodstream can escape into the extracellular matrix and enter cells, contributing to their stress and disorganization, and other materials can escape from cells and enter the bloodstream. One of the simplest demonstrations of fibrin leakage is to shine a beam of light into the eye. The presence of fibrin and other inappropriate molecules diffuses the light, causing a quote-unquote flare in the aqueous compartment. Albumin, a small protein from the blood, is often seen in the urine during stress. The effects of that sort of leakage vary with each organ. So it sounds like, for example, if you're driving and, at night and you have all of these headlights coming towards you and they are causing that flare that a lot of people experience when driving at night. Maybe that is an easy indicator of this leakage issue. Fibrin is an essential structural and functional part of the organism, but when it escapes from the bloodstream, it participates in the degenerative processes of inflammation, fibrosis, and tumor formation. Its fragments stimulate secretion of inflammatory mediators, and that's from a study by Hamaguchi et al. 1991. In the hormonal environment dominated by estrogen, mild stresses such as exertion or even restless sleep allow toxins and sometimes bacteria from the intestine to enter the bloodstream, triggering a complex chain of events that create a systemic inflammatory state. Although these processes have been observed in many simple experiments, their implications are almost always neglected or denied or explained away. Incorporation of certain polyunsaturated fats into the tissues increases the leakiness of blood vessels and amplifies the reactions to stresses and inflammatory stimuli. And this is why I talk about it taking time for all of these lifestyle, environmental, nutritional changes to take effect, right? Because these polyunsaturated fatty acids have integrated themselves into your body, and it's going to take a while, right? It says incorporation. It's going to take a while to unincorporate, as it were. Antioxidants, thyroid hormone, progesterone, and anti-inflammatory agents, including glycine or gelatin, niacin, and I know Dr. Peat is pretty stickler on the use of niacinamide, so I'm surprised to see him say niacin here, because there's a lot of different forms out there, and he says the niacinamide version does not have the inflammatory issues that other versions, like slow release versions, might have. And saturated fats can prevent and in many cases reverse these degenerative inflammatory processes. Even a single-celled organism has to keep its parts separate, and highly differentiated multi-celled organisms have many special systems that serve to keep their parts separate, so the different tissues and organs can maintain their distinct functions. The movement of substances from blood to cell and from cell to cell is normally very tightly controlled. And when the systems that control those movements of water and its solutes are damaged, the tissue's structures and functions are altered. Okay, so when it says water and its solutes, solutes are just things dissolved in that water, right? That's what a solution is. You have your solvent, which is the water, and then you have your solute, that which is dissolved in the water. The prevention of inappropriate leakiness can protect against the degenerative processes and against aging itself, which is among other things a state of generalized leakiness. That's good. I need a shirt or a bumper sticker that says opposing generalized leakiness. When cells' energy is depleted, water and various dissolved molecules are allowed to move into the cells, out of the cells, and through or around cells inappropriately. Right? This is when the energy is depleted. The weakened cells can even permit whole bacteria and similar particles to pass into and out of the bloodstream more easily. And this is the problem with most people's framework for their health and nutrition, is that they don't have a framework, right? And so a lot of what Dr. Peat's framework is based around is the ability to create energy. Okay, and so this is why a lot of people in this sphere consider themselves what you might call pro-metabolic. Metabolic implying metabolism, right? The creation of energy in an appropriate way, in an efficient way. Okay? And so you'll find a lot of these issues start happening when you cannot create energy efficiently, or when your energy is depleted quickly, things along this line. And that's extremely simplified, obviously, but it's good to have a framework to base things around, because then otherwise, you know, you're just grasping at straws, however they say that. And someone comes on the social media with a quick 30-second reel, and it's all catchy and all this, and you grab onto it because, you know, of how they look and they sound confident, and in reality it can be hurting you more than helping, right? Not to insult people's intelligence, but you know, it is easy to be influenced if you don't have a framework, if you don't have a foundation, right? One of the earliest investigators of the effects of stress and fatigue on nerves and other cells was A.P. Nasonov. In the first half of the 20th century, A.S. Troshin, 1956, has reviewed his work in detail. He showed that in cells as different as algae and nerve cells, fatigue caused them to take up dyes, and that the dyes were extruded if the cells were able to recover their energy. When nerve cells are excited for a fraction of a second, they take up sodium and calcium, but quickly eliminate them. Prolonged excitation leading to fatigue can gradually shift the balance, allowing more substances to enter and stay longer. When nerves or other cells are quickly killed by heavy metals, such as osmium, the metals are visible in a layer at the surface, which is sometimes taken as evidence of a quote-unquote cytoplasmic membrane. But if the cells have suffered oxygen deprivation or have been injured by X-rays, the metal will be visible as a gray color evenly distributed through the cell. The deposition of the metal occurs when it reacts with electrons. in the relatively vital cell, the heavy metal stops at the surface and is mostly reduced there. But the devitalized cell presents no structural or chemical barrier to the entry of the metal, and the reactive electrons appear to be evenly distributed through the cell. Oxygen deprivation, X-irradiation, and other stresses cause the cell to be unable to use electrons to produce energy, and instead the electrons are available to react destructively with whatever may be available. While Nasonov showed that dyes and even particles enter energetically depleted cells, newer techniques are able to show that the leaky cells are structurally disrupted by the excessive reduction of their proteins by excited electrons and free radicals. Okay, so to break that down, you either have a cell that is utilizing electrons appropriately, right? In the mitochondria, they're passing electrons along the electron transport chain to create energy. Or, like it's saying here, you have this excitation happening with these, right, excited electrons, free radicals. Two different things that can happen, right? Things can be reduced or things can be oxidized. When something is oxidized, you have an electron acceptor, such as oxygen, and it accepts electrons, okay? It removes those and accepts those. Whereas, if something is going to reduce something, it's going to provide electrons to it, right? And so again, you have these excited electrons, free radicals, that are attaching themselves, right, reducing the proteins. Okay, and so you can imagine if you're not oxygenating your cells, if you're not well oxygenated, which requires CO2, then you don't have oxygen there to accept these electrons. Okay, and you have other stressors that can cause, excited electrons, like it's saying, which again, this is the cascade of events that happens in, a poor environment, essentially. Moving on. In the 1970s, experimenters found that mussels from vitamin E deficient animals released their enzymes when washed in a saline solution more easily than did the mussels from vitamin E replete animals. And replete just meaning abundantly supplied, right? The opposite of depleted. Replete. Other experiments around the same time showed that reducing the ATP of mussels caused a similar loss of their ability to retain their proteins. Over the years, many experiments have established, both in vitro and in vivo, okay, and in vivo obviously being more helpful because vivo in Latin meaning life, so this is like in a real biological sense. Whereas in vitro, vitro actually means glass, so this is talking about outside of its natural biological state in a petri dish, right? Over the years, many experiments have established both in vitro and in vivo that fatigue, stress, aging, and inflammation cause cells to lose their normal constituents, but also to allow foreign materials to enter more easily. When I was working on my thesis around 1970, investigating the effects of aging on the metabolism of the uterus, I found that the changes occurring during aging were, in all the ways I tested, the same as those produced by X-irradiation. Excess estrogen, oxygen deprivation, excess polyunsaturated fats, and vitamin E deficiency. Although everyone working in the lab was familiar with the appearance of the uterus from old hamsters, they are typically large, stiff, and bluish. Everyone was surprised when I suggested that the aged uteri seemed to function as if they were under the influence of a considerable amount of estrogen. Everyone was familiar with the medical textbook doctrine that, quote, menopause is caused by estrogen deficiency, end quote. In humans, gynecologists know about, quote-unquote, Chadwick's sign. the fact that the uterine cervix turns blue or purple during pregnancy, and everyone knows that blood is blue when it's deprived of oxygen. So it's surprising that estrogen's effect on tissue oxygenation isn't widely recognized. When estrogen is given to an animal, it almost instantly causes capillaries to become leaky, allowing water to move out of the bloodstream, and at the same time, estrogen causes cells to take up water, right? Edema. Both of those processes are the same as the early effects of oxygen deprivation. In the normal reproductive cycle, the surge of estrogen lasts only a few hours, and normal permeability is quickly restored by increasing progesterone. During those intermittent, short exposures to estrogen, there isn't a massive leakage of serum proteins into the tissues. During the time of estrogenic influence, all kinds of cells are influenced, with the excitatory equilibrium of nerve cells, glandular cells, and immune system cells being shifted, lowering the threshold of excitation or prolonging the excited state. Anything that causes inflammation causes a similar loss of water from the blood as it is taken up by swelling cells. If inflammation is generalized, it causes circulatory shock because the volume of the blood has become insufficient to serve the organism's needs. One of Hans Selye's earliest observations of the effect of an overdose of estrogen was that it causes shock. Although water loss causes the blood to become more viscous, okay, meaning you can imagine molasses, and when you put it in the fridge, it's very viscous, right? There's like friction between the molecule. It pours slowly, right? Molasses obviously being more viscous than water, right? Okay, so although water loss causes the blood to become more viscous under the influence of estrogen, right, so stickier essentially, the plasma becomes hypotonic, meaning that it contains fewer osmotically active solutes than normal. Okay, hypotonic. Some of the sodium that helps to maintain the blood's osmotic balance is lost through the kidneys, and some is taken up by the red blood cells and other cells. The osmotic imbalance of the blood causes tissue cells to take up more water, contributing to their increased excitability. In many cases, the vascular leakage of inflammation and shock can be corrected by using osmotically active substances such as starch solutions, gelatin, or concentrated sodium chloride, meaning salt. And I bring up Dr. Tom Brewer, as does Dr. Peat bring him up. I actually was introduced to him before Dr. Peat, and Dr. Tom Brewer has what's called the Dr. Brewer pregnancy diet. You can look it up online, and it's absolutely amazing, doing wonders for women as they prepare for pregnancy, through pregnancy, and afterwards. But anyway, they have a strong emphasis on salt, and so this is kind of a primer in Dr. Brewer's research here. And anytime Dr. Peat brings up salt, he tends to reference Dr. Brewer because it's Dr. Brewer's forte. Moving on, the tissue water retention caused by estrogen, hypoxia, and stress is analogous to the swelling of gels and colloids. That is, it's governed by the state of the electrons and counterions in the system. Excitation, fatigue, or injury can cause a shift of pH toward alkalinity, causing water uptake and swelling. And there's so much to say about the whole trend out there to eat alkaline and these things of this nature that just don't make any sense. There's not any physiological thought that goes through these concepts of eating alkaline or eating acidic. There might be like hints of truth throughout that, but just quote-unquote eating alkaline and what people consider that to be and the foods that they consider alkaline, it's just completely lacking any type of evidence. Our body has different locations that are alkaline and acidic and things of this nature. So anyway, The blue color of the pregnant cervix or of the uterus in an animal given an overdose of estrogen indicates that the tissue isn't sufficiently oxygenated to maintain its normal red color, even though the flow of blood is increased. Some experimenters have noticed that newborn animals sometimes have the postural reflex, lordosis, that indicates an estrogenic state, and that suffocation can produce the same reflex. Irradiating animals with X-rays will also produce the whole range of estrogenic effects. That really is interesting. Kind of a whole weird rabbit hole to go down, the lordosis reflex. But anyway, it's the lordosis reflex is kind of the bending of the tail end of the spine in a female animal, and this happens in animals. You see this when they are preparing to be mated. And that is interesting that that's associated with oxygen deprivation and also high estrogen, right? Because high estrogen, your highest amount of estrogen, naturally, optimally occurs like the days before ovulation, right? When you are preparing to create a baby. Anyway, again, a whole rabbit hole to go down, but very interesting that he is connecting those. One of the features of the aged uterus that I studied was the age pigment lipofuscin, a brown, waxy material that accumulates in old or stressed tissues. Prolonged dosage with estrogen accelerates the formation of this pigment, which is largely derived from oxidized polyunsaturated fatty acids. And not a good thing, right? Increased amounts of those fats in the diet or a deficiency of vitamin E, because vitamin E is protective against those, or exposure to ionizing radiation or oxygen deprivation can also accelerate the formation of the age pigment. The presence of the pigment intensifies the effect of estrogen, since the pigment wastes oxygen by functioning as an oxidase enzyme. Other tests that I did on aged or estrogenized uterine tissue indicated that several oxidative systems were activated. For example, the tissues showed an extremely high activity of the enzyme peroxidase and a very intense reduction of a chemical dye, tetrazolium/formazine, that indicates the presence of reductive and oxidative activity of the sorts caused by radiation and oxygen deprivation. These reductive and oxidative processes include the production of some free radicals that are capable of reacting randomly with polyunsaturated fatty acids. The interactions between estrogen and the polyunsaturated fats are now coming to be more widely recognized as important factors in the inflammatory, hyperpermeable conditions that contribute to the development of heart and blood vessel disease, hypertension, cancer, autoimmune diseases, dementia, and other less common degenerative conditions. Estrogen increases lipid peroxidation and maintains a chronically high circulating level of free fatty acids, mainly PUFA, activates the phospholipases that release arachidonic acid from cells, leading to formation of prostaglandins and isoprostanes, and increases the enzymes that form the inflammation-promoting platelet-activating factor, while suppressing the enzymes that destroy it, and increases a broad range of other inflammatory mediators, interleukins, and NF-kappa B. All of this caused by estrogen. Okay? If you want to cause anxiety to someone who's in the pro-metabolic community and reads these things often, just read them that paragraph right there. You're going to give them nightmares. Okay. All right. The leakage of enzymes out of cells and into the bloodstream is recognized medically as evidence of damage to the organ that is losing them. Different combinations of enzymes are commonly considered to be evidence of a heart attack, or skeletal muscle damage, or liver disease, pancreatitis, prostate cancer, etc. But often the reason for the leakage isn't understood. Hypothyroidism, for example, causes leakage of enzymes, possibly mainly from the liver, but also from other organs. Excess estrogen, intense exercise, starvation, anything that increases lipid peroxidation and free radical production, such as drinking alcohol when the tissues contain polyunsaturated fats, can cause organs such as heart and liver to leak their components. The loss of enzymes increases the energy needed to stay alive, but it doesn't necessarily change the basic functions of the cell. Though, when mitochondrial enzymes leak out into the cytoplasm, the cell's energy metabolism is impaired, at least temporarily. But the entry of catalytic materials from other tissues changes the organization of a cell, giving it conflicting instructions. In many situations, as L.V. Polizhev and V. Filatov demonstrated, the substances released during stress and degeneration serve to stimulate healing and regeneration. But when the resources aren't available for full repair or regeneration, only a scar or atrophic fibrosis or a tumor will be formed. In severe stress, intracellular fibrin deposits have been found in the heart and other organs, including the prostate gland. Deficiency of testosterone causes vascular leakage into the prostate. Interesting. Fibrin promotes tumor growth, partly by serving as a matrix, partly by releasing stimulatory peptides. Kidney disease, diabetes, pregnancy, toxemia, and retinal degeneration are probably the best known problems involving vascular leakage. But increasingly, the big C word and heart disease are being recognized as consequences of prolonged permeability defects. Congestive heart failure and pulmonary hypertension commonly cause leakage of fluid into the lungs, and shock of any sort causes the lung to get quote-unquote wet, a waterlogged condition called quote-unquote shock lung. Simply hyperventilating for a couple of minutes will increase leakage from the blood into the lungs. Hyperventilation decreases carbon dioxide and increases serotonin and histamine. And I was just reading about this. Dr. Peat, in an interview I was listening to, talked about a negative ion generator being one of the best ways to decrease serotonin while you sleep, and something about how it helps the lungs essentially detox from serotonin through the night. So something to look into. We've been using a negative ion generator in the home for quite a long time now, and when I first got it, probably the most obvious beneficial thing that I've done health-wise for a very long time. Immediately felt like I was just breathing outside because of how clean the air felt. Hyperoxia itself contributes to lung injury and exasperates emphysema, though it is common to see patients breathing a high concentration of oxygen. Emphysema, which can be caused by hypothyroidism or hyperestrogenism, and often can be cured by thyroid or progesterone. And many other respiratory problems are associated with capillary leakage. Cells of the lung and intestine are able to synthesize their own fibrin, apparently because of their special problems in preventing leakage. Prolonged systemic inflammation can lead to lung fibrosis, and fibrosis increases the likelihood of lung cancer. The inflammatory state that causes exaggerated cellular permeability is very closely related to quote-unquote hyperventilation, the loss of too much carbon dioxide. And this is why I always talk about the Buteyko method. Just look it up, and it's easy things you can do throughout the day in order to increase the amount of carbon dioxide in your system. The release of serotonin during hyperventilation isn't the only cause of vascular leakage. The carbon dioxide itself is an essential factor in regulating the state of cellular electrons in maintaining cellular integrity. Hyperventilation, like the shift from oxidative to glycolytic energy production, right? That's a bad thing. That typifies estrogenized or stressed cells, or the big C word, raises intracellular pH. In the case of mast cells, increasing alkalinity causes them to release histamine. And there's a study. But similar alkaline-induced exocytosis seems to occur in all stressed tissues. The blood platelets that become incontinent and leak serotonin in the absence of carbon dioxide are undergoing the same structural stresses experienced by endothelial cells, smooth muscle cells, mast cells, and other cells when carbon dioxide is depleted. Although it has been about 70 years since Yandell Henderson made it clear that supplemental oxygen should be combined with carbon dioxide, mechanical ventilation in hospitals is still causing lung injury resulting from hyperventilation, i.e., the absence of carbon dioxide. A similar misunderstanding of biology was involved in the use of dialysis to treat kidney disease. Until recently, commercial dialysis fluids contain acetate and/or racemic lactate instead of bicarbonate, because of the difficulty of preparing bicarbonate solutions. And the result was that very prolonged dialysis would damage the brain and other organs. And there are some studies there. Dialysis has been seen to increase lung permeability. And that's from Bell et al. 1988. Amyloidosis produced by chronic dialysis affects all organs, but its effects are best known in the brain, heart, kidneys, and lungs. Serum amyloid A is one of the acute phase proteins, like C-reactive protein, or CRP, that are produced by inflammation. Estrogen, radiation, and other stresses increase those pro-inflammatory acute phase proteins and decrease protective albumin, which is called a negative acute phase protein, since it decreases when other acute phase proteins increase. The liver is the major source of the acute phase proteins, and it is constantly burdened by toxins absorbed from the bowel. Disinfection of the bowel is known to accelerate recovery from stress. And we know that Dr. Peat talks about the carrot salad, the raw carrot salad, well-cooked white button mushrooms, bamboo shoots, and activated charcoal. And then, of course, the smart and calculated use of particular antibiotics. So a lot of different ways that you can, unburden the bowel, the liver. Seen from the perspective of the stress leakage syndrome, any serious injury or sickness damages all organs. The exhaled breath is being used to diagnose inflammatory lung disease, since so many of the mediators of inflammation are volatile. But systemic diseases such as the big C word and arthritis and relatively minor stress can be detected by changes in the chemicals found in the breath. Polyunsaturated fats and their breakdown products, aldehydes, prostaglandins, isoprostanes, hydrocarbons, and free radicals, and carbon monoxide, nitric oxide, nitrite, and hydrogen peroxide are increased in the breath by most stresses. Both proline and glycine, which are major amino acids in gelatin, are very protective for the liver, increasing albumin and stopping oxidative damage. And so I have a really sensitive carbon monoxide detector. And so anyway, I'm interested if it would be able to pick up any amount of carbon monoxide being breathed out. I've tried it. I've never had anyone trigger anything over 0 parts per million. But and then it'd be really cool to get a nitric oxide detector, because I think that would probably provide a better, more easily detectable indicator of stress on the body via, breathing onto it. Saturated fats are protective against free radical damage and can reverse liver fibrosis. Thyroid hormone protects against excess estrogen and can prevent or reverse fibrosis of the heart. Anti-estrogens are widely effective against vascular leakage. Thyroid, progesterone, and testosterone are among the most effective natural anti-estrogens, and they are curative in many conditions that involve vascular leakage. Progesterone and pregnenolone have been called the anti-fibromatic steroids, and it has been used to treat many inflammatory and fibrotic diseases, including the big C word. The anti-serotonin drugs are being increasingly used to treat fibrotic diseases and other problems related to vascular leakage. Anti-inflammatory and anticoagulant things, especially aspirin and vitamin E, protect against the accelerated turnover of fibrinogen/fibrin caused by estrogen in the various inflammatory states. All right, that article, feel like that's just filling a lot of gaps in how different things are connected. That really brings a lot of these different topics together. Anyway, very much appreciated that article. Hopefully you guys got something out of it. Please like the video, subscribe to the channel, share this with the people who need to hear it. Until next time, God bless.